the review on mitosis. Mitosis is a process of cell division, and in this cell division it's designed for growth. As we take a look at the lecture notes today, we'll notice that we have a lot of keywords here. Chromatin. Chromatin is mass of DNA and protein that makes up chromosomes. And to me it looks like an unwound baseball with a bunch of the yarn inside and the thread inside just completely untangled and loose. Chromosome. Chromosome is another key term that we are going to be using. They're tightly coiled mass of DNA and protein. The centromere. Centromere is one key term that we're going to use to just kind of focus on main points of interest, especially during the different phases of mitosis. And then finally the chromatid. The chromatid is one of two identical strands of DNA that make up the chromosome, especially in the typical view of the typical X pattern that you see most of the time when you see mitosis drawn. On the next page we notice that we've got homologous pairs as well. Homologous pairs, chromosomes that possess the same genes but not necessarily the same of each gene. Now, we'll go ahead and get started today and start talking about the 6 phases. The phases of mitosis, they are designed to show a chronological order of events in the replication of DNA as well as the changing and reproduction of a new cell. As we look at over here in the corner, I'm going to show you very quickly. Let me select this first. As we look at this little dot that we see right here, and as I select this, you're going to notice that as I pull this outward, you can see the chromatin as it is expanded. Now as we scroll it in, we notice it's very, very tiny inside the nucleolus of the nucleus. And it's a very tiny, small little speck, and it is expanded. Moving to the next phase, we're just going to look at the very first phase, and that's interphase. A lot of the process, a lot of the duplication takes place in interphase. We see the chromatin in full force at this moment, and we see that all the chromatin and all the strands of the chromosomes are starting to duplicate and make pairs. Now, for animation purpose, what we have to do now is, as I click each one of these, we can see in interphase we start to get the coiling of the strands. The strands will start to come together, and as they come together, the strands will wind up nice and tight to where we see that homologous pair look and that typical look that the chromosomes have, which is in that X factor. Now, looking at this point right here, we can see where I'm marking on the blue here, we see that that's the centromere, and all these have a centromere. All of these are going to be that's the middle, that's that bridge that puts the two chromatids together. And as I label this one here 1 and then this one 2, we notice that this chromatid is the exact same as this chromatid, and they are exact pairs. They are copies, as we see that. Now we move into the second phase of mitosis, which is prophase. Now in prophase, we notice that these two little spindles start to appear, and these are going to be very important in the pulling of the cell apart. At first, all the paired chromosomes that are all copies, they will all start to move towards the center, or the equator in this instance. Now we notice as the spindles move away from one another, we see that the spindles start to reach out and almost get like a web of some sort, you know, and these spindle fibers is what they're called. And these fibers will stretch out across the cell and grab a hold of the centromeres of these chromosomes. And we're still in prophase. Now, getting really close to the end of prophase is when the chromosome pairs line up on the equator. Now, I say that word "on" because that is a huge word in this whole process. In meiosis, you'll see that we will use the word "at the equator" because there's a little difference, and that's probably the main difference between the two, to give different variety or keep the same cell. So as we go to the next slide here, this is metaphase. Now we're in the third phase. Metaphase, the way I remember metaphase is meta is in the middle. So the M's are in the middle, and all the centromeres, as we can see here, are all lined up. They're all lined up on that equator. And we think of the equator as this imaginary line that runs right through the middle. And there's that imaginary line that we see running right through the middle. And those centromeres are all lined up. And notice, number 1 chromatid is over here, and number 2 chromatid is over here. And as we see that, this is one pair and this is another, and these two are the exact same. They're exact same. Now, at the end of metaphase, we see the spindle fibers have totally attached to all ends and the centromere of the chromosomes. And as they are attaching to the chromatids, they are going to then reach the next stage, which is anaphase, which is the result of pulling them apart. As they pull and break the centromere apart, the two sides now start to pull apart from one another to the point that it creates a force of the chromatids going back towards the spindle fiber, as I show in this direction here. As I draw that arrow, they're going in this direction, this one's going in this direction, and in doing so, we will start to see at the top here, we'll start to see kind of a breaking in the cell as we see it up in here as I'm locating. But we're going to see that, and that causes the cell starting to split. And notice once we see that, we are now in the final stage, which is called telophase. Now, telophase, not much is going to go on in telophase, except for the cells do split. As we see it here, we see the equator line, the equatorial line is now splitting into two, and the spindles now will start showing that the nucleolus is going to come back and take over, and then nice and neat, the chromatids are in the cells. And if we take a look at it, we notice we have one green, two green, and we have one green, two green, we have one yellow, we have an orange, red, blue, and back here behind we see a blue. Not very good graphic there. And then we have another blue here. So we do have identical cells now, and it's all because of the duplication of the chromosomes early on in the entire process. Now that the nucleolus is back, the nucleus is back, we see that the chromatin, or the chromatids, unwind into that chromatin to where now we see it slowly get back to where its original location is. And again, as we look at it, we see that this here is, these are two identical cells. And the purpose of this is for growth. These are skin cells, these are body cells, these are cells that are going to continue to regenerate from the existing cells. And that leads us to what we talked about the other day in history when we look at the cell theory. And the third rule of the cell theory is new cells are produced by existing cells. And from the one existing cell that's already there, we have new cells produced. And because those two cells are now two identical cells, those two cells can go into mitosis again and start it all over again. And